This episode of TWiM is sponsored by ASM Gap, and they want to tell you all about an ASM workshop called Turning Your Science into a Company. This is running on October 1st through 3rd, and the application deadline is August 20th. Have you ever thought about taking what you do in the laboratory and making a company out of it? Well, that's what this conference is all about. It's going to teach you how to do that. And they'll have successful entrepreneurs who have done exactly that. They've started companies based on scientific discovery. So some of the participants in this meeting will be people who have done that, and you're going to get practical tips, advice, and resources on how to establish a startup. So if you ever thought about it, but you never really could figure out the next step, whether you're an early, mid, or late career person, it's a 2.5-day entrepreneurship workshop. Basic information on making a startup. You'll get information using examples from companies that are involved in the industry. Day 1, planning a startup. Day 2, executing the plan. Day 3, exiting the business. That means selling it so that you can benefit from what you have done. And this is good for an advanced graduate student, a postdoc, early career scientist, wherever you are. Why not advanced career scientist? If you want to do something different, why not? Sounds like a really good conference. So again, you can find the information at ASMGAP, G-A-P, asmgap.org/TSC. The deadline for applying is August 20th. Check it out. This week in microbiology, episode number 108, recorded on July 16th, 2015. Hi, everybody. I'm Vincent Racaniello, and you're listening to TWiM, the podcast that explores unseen life on Earth. Joining me today from Small Things Considered, Elio Schaechter.
Well, hello there. I know I sound bad.
That's okay.
That's not me. It's not me. I'm sorry. I apologize.
Yeah, well, you have the aftermath of a cold, plus...
and my computer has the aftermath of something, so...
Yeah, your mic isn't working, but I'm sure people will still be happy to have you here.
Thank you. Let's hope so.
Also joining us today from Ann Arbor, Michigan, Michele Swanson.
Hello.
Haven't spoken with you in a few weeks.
I'm back.
Are you having a great summer?
Yeah, I really am. It's been so beautiful here in Michigan.
Have you been golfing at all?
I sure have.
Great. Good to hear it. I have not done any golfing.
I scored a personal best the last time I was out, 42 for 9 holes.
Pretty good.
Wow.
I'm happy.
Fantastic.
That would be 84 for 18 holes?
Yeah, assuming you could be consistent over 18 holes.
Because you would get tired and probably start messing up, right?
Or sharper and more focused. Who knows?
Also joining us today from Charleston, South Carolina, Michael Schmidt.
Hello, everyone.
How have you been?
I've been good.
All right. I heard about you from my colleague Paula Trackman.
Yes.
I guess there was a meet and greet, and you were there, right?
Yes, she is now officially ensconced as our new dean of the College of Graduate Studies, and she's full of new and exciting ideas and wants to do all sorts of great things. So I'm always happy to help out, and I'll be one of her co-conspirators.
Speaking of new and exciting things, that's exactly what we have for our listeners today. we have 2 things we're going to talk about. We're going to talk about a paper, but before that, we're going to talk about what's not a paper. Well, it is a paper, but it's not a research paper. Or maybe it is, but not experiments in using small volumes of liquid, let's say that. It's a study out of the University of Wisconsin-Madison. And I'm going to hand it over to Elio, who's going to go through it, and I'm sure we're going to all chime in at one point or another.
So, okay, well, the paper is entitled Strategies from the University of Wisconsin-Madison for Rescuing Biomedical Research in the U.S. It was published in eLife, which is a new e-magazine, and it has 17 authors, so I won't tell you who they are. I recognize a couple of them. Anyhow, what this is the report based on a cross-campus study, which was also not just cross-campus, but cross-career stage, involving senior people, mid people, junior people, which went all the way down to graduate students, and cross-disciplinary, namely all kinds of fields of which are impacted by the funding in biomedical research. And what this is another one of several pieces of writing that have emerged about the disastrous situation of the last 15 years regarding the funding of biomedical research and the shape of the biomedical research institution. As we all know, I'm sure most listeners know, the degree of funding has gone down so significantly that in a period of 10 years or so, the success rate of people who apply for a grant went down to a low of 30% to a non-sustainable, abysmal 15%. And what has happened is that people have to spend a significant amount of their time, maybe a third of their time, maybe half their time, writing grant applications. In addition, a number of other things have happened. Money has been shifted around from the traditional single research lab investigator to large groups or to laboratories which are not just composed of an investigator and a handful of acolytes, but very large labs. And from research grants, funds have been sifted off to project grants, to contract grants, to other things, all of which has conspired to make this into a disastrous situation. That is, if you believe that the key to biomedical research is the single investigator lab.
But it's not all bad, because some of those initiatives have been things like funding the Human Microbiome Project.
That's right.
And so there have been some good initiatives that have really helped to propel our discipline forward. Sure. But at the same time, it's, as Elio's just about to tell us, it's getting worse.
Well, it's so bad that something has to be done. Now, the good news is when you read the papers is that the House, I guess the House passed a bill which increases the NIH budget substantially. There's other issues which we won't go into, having to do with the FDA. Won't go into that. But there is some hope on the horizon of restabilizing this, because this was really an event that was unprecedented. In my lifetime, which is now going back to the early 1950s, the funding by NIH and NSF, government funding of research, has oscillated. It has waved up and down with, there's been a way of life. We could adapt to it to some extent, but in recent years, this became disastrous. And of course, included in these various new ways of funding has also been one of the overwhelming facts, namely that the number of people being trained has increased very, very much. And so the denominator of people applying for a grant has increased hugely, and the denominator has stayed, the people who receive grants has gone down. So all these factors have conspired. And the good people at Wisconsin made a study of this. It's a very rational study, and I think it's said beautifully. I recommend this to all. You don't have to agree with everything. You may not even agree with some of the major conclusions. But may I go over a little bit how this paper is structured and what it says?
Yeah, sure.
Okay. So the main conclusions is that there is a need for overhauling the system and that funding needs to be increased. They found a number of themes which we all can agree with, namely that science is essential for the well-being of this planet, that research is the responsibility of individual investigators, and so forth. It goes on and on. There's about 9 or 10 themes of this sort. Let me single out 2 that I like especially, namely breakthroughs in science can't be planned. That's true to some extent. There are some things which you can, like what Michael just referred to, the study of the microbiome, is something that lends itself to planning. But a lot of breakthroughs that happen are either serendipitous or whatever they are not something that could be written down in a grant application that people would like to read.
Because it wouldn't get funded. And, that's part of the partial problem is if you write risky science, or you write for breakthroughs, you won't get funded.
So given these facts, they point to a number of symptoms. They don't call them the cause, they call them the symptoms of the condition. And namely that they're the following. NIH reviewers are overwhelmed. The reason is that the number of grant applications has increased hugely. It used to be that you wrote a grant application, you had a reasonable expectation to get it. Then it got worse, so you wrote 2 grant applications. Now you write what? 5, 6, 8 grant applications. Somebody has to deal with them and usually read them. So the system is overstretched way beyond reasonableness. In addition, they say a symptom of the condition is that it reduces the diversity of research. That is, people go for the safe stuff, like Michael just said. You don't get funded for a great idea that's far out. There's inefficiency in the system because of the instability. That is, if you can't plan efficiently. And lastly, and that we just already said this, too much time is spent writing grants. These are symptoms, okay? They make a bunch of recommendations, and I would like to tell you what they are. The recommendations are, some, as I say, are controversial, some are not. One is a large number of grant money goes to funding individuals' salary. That is the salary of the principal investigator, not the salary of the technician or the student of the postdoc. That's something else. But the salary of the principal investigator is often handed over to the granting applications as part of the grant, and it becomes a substantial portion of the grant money. So a lot of money goes to that. So they recommend to limit salary of PIs to 50% of the salary. Okay.
But at the same time, you also have to point out that those salary dollars also incur overhead. And overhead is often 50 cents on the dollar.
Oh.
And so not only are you paying for the salary, you're paying an additional 50 cents. And in reality, and this is one of the conclusions that they don't touch in their discussion, is that should there even be overhead on the PI's salary, because all they do is literally tick a computer box on a screen to change your payroll from the university to a different funding source, and they do it but once for the entire period of the grant. So why does that cost them 50 cents on the dollar that they're already paying you? So that's one of the gaps that they left out of their discussion, and it may be because indirect costs are the sacred cow of universities.
But I don't think these people minded goring sacred cows, so you can't accuse them of that. The thing is that the document cannot deal with every aspect of it.
That's true.
Right. This is an important point. The next is that they would like to shift back a higher proportion of money to individual investigators and away from projects. Like Michael just said, some projects are terrific, should be funded, but are probably too many. Thirdly, they suggest that laboratories should be limited in what the total amount of grant money is that they receive. This is extraordinarily controversial because a lot of people need a lot of money because they run a very large operation. They come up with a figure of $1 million a year per laboratory. That's a lot of money for some. That's no money at all for others. On the other hand, they don't do away with the idea of contracts and projects and so forth, so that there could be, for the people who have to have more money than $1 million, there are other avenues. These are the main recommendations. Now they go on and deal with further problems. One of the problems is that there are too many postdocs for the available academic positions. Okay, this is a huge problem. A lot of postdocs come to your lab, a research lab, desiring to follow in your footsteps. That is, you are a professor, they want to be a professor. The fact is that there is just nothing like the number of available jobs, no matter how much you extrapolate into the future, for all the people who would like a job. So they would like to cut down on the number of postdocs, not the number of graduate students, but the number of postdocs. Cutting down on the number of graduate students was proposed by other people who recently wrote papers of the sort. So they would like to cut down by making it possible to get a master's degree that is meaningful and which is respected. You see, a lot of people could contribute to science very well with a master's degree, and that should be encouraged. I believe that maybe that is happening to some extent. They also like the idea, this is probably a side point, of staff scientists, that is, people who are in a laboratory as lieutenants to the boss. Anyhow, I won't go into that. There's another one which I like a lot, and I wonder if we should spend a little time on it, is to increase incentives for retirement. And here I'd like to take a second and quote from an email of our colleague, Roberto Coulter. Roberto is a professor at Harvard University, and for those who don't know him, he's one of the stellar contributors to modern microbiology. His lab has been exceptional. And he is, I don't know exactly what his age is, but he must be in his early 60s, and he's considering retirement sometime between mid-2017 and mid-2018. He's not taking any more postdocs, and his last NIH grant will run out then. So 2 or 3 years from now, the Coulter lab will close down, and he will retire and do other things. So let me say what he calls the reason. Lots of reasons. One important reason is that he believes that as a community of scientists, we're training way too many people. At the current rate of expansion, we all know the system is not sustainable. Again, I'm quoting. But many of my colleagues, like ostriches, pretending this is not an issue. About half the people who come to my lab have come in the hope of becoming, like me, heads of research labs at universities. Over 33 years, I've placed about 50 people in faculty position. If each of those 50 train 50 new professors in the next 30 years, can research funding really keep up with that insane exponential growth? Of course not. So we need to slow down this insanity. Once more ways to help is for people like me to retire on time. So he says, retiring now is the only responsible thing to do. I see some of my colleagues 10 to 15 years, my seniors still having labs of 10 to 15 people. That's just one reason. Okay, so that I asked Roberto if it's okay for me to quote him, and he said it is, so I just did. So this is one of the recommendations and one of the things that may have an effect. So this is more or less an overview, an idea of what this document is about. There's quite a bit more in it, and tell me what you think.
Well, I want to just mention that some of the things they also talk about have to do with grant submissions and grant applications and grant review, and they're very good because I think that's a big part of the problem. I don't think we don't have to go over them. This is an open access article, but they'd like to reduce the number of grant applications, reduce the pay line, or fix the pay line, make higher standard peer review. And here's one that I really liked. We encourage the NIH to bolster recent efforts to eliminate the use of translational relevance for judging basic science proposals, which I think is really good. So these are all important components. I mean, what you've mentioned are really good as well. But fixing the grant application process is also part of it. Now, I think that I just had a few comments because we've discussed this also on TWiM because there have been some other papers, notably the Alberts paper in 2014 discussing this. And the idea of limiting faculty salary reimbursement is great. This is going to save a lot of money, but this will require the universities to pay their faculty as they should be paid. Because right now many universities are getting free rides. And this is not just for salary, but for indirect costs. And it's not something you can change overnight, but it has to be fixed. And that would really go a long way to fixing the shortage of NIH dollars. I don't know how you're going to do that. And I think that's a long-term solution because universities are going to have to come up with the money to take care of that. And as we know, that's not an easy thing to do. But I think that's a crucial part of it as well.
I think one result would be that the university departments in science would get smaller. I mean, they won't. Look, every university, every research university I know would like a number of members of the National Academy of Sciences on its faculty. Everybody's ambitious. Everybody wants the best. Well, there's a limit to that. There's a limit. You just can't do it. Everybody can't be the best.
Well, not everybody has to be an academy member, but, great universities are great because of their faculty. So, for a great undergraduate education, is really enhanced by a great research faculty, or I would say even made by a great research faculty. So, you make it small. There are lots of small colleges that have good programs, but, you could argue that it's really nice to have a large, diverse research faculty.
It's wonderful. It's wonderful, but you can have everything you want.
Mm. Well, if
you just have to figure out how to afford it.
They have to pay for it. And right now, the universities aren't paying enough. Right? I just think 50% is a good compromise, and indirect costs have to go down as well. That would really help.
And they haven't explored the discussion about indirect costs to the level and extent, because computer systems have advanced substantially in the last 20 years, and it really doesn't take as many dollars as the indirect costs were originally assigned to. The universities are using the indirect costs for the unfunded mandates that the federal government has imposed on them to do other things. And so the indirect costs are often spent many times over at universities. And so they argue that those indirect costs are the mother's milk of the institution because they couldn't survive without them, when in reality, it's the investigator that is generating the indirect costs, and they aren't seeing the value associated with those indirect cost dollars in the sense of, aside from the utility bills that the indirect costs are paying for, they often don't appreciate or understand where their dollars are going.
I think you're right, but I think we should leave the subject of indirect costs aside.
Yes.
One, but there are many others. So let's talk about something else.
Michele, any thoughts?
Yeah, I actually, this is a topic that I've thought about a lot, and I should say that here at Michigan, we recently hosted a national conference of people from more than 120 different institutions, public, private institutions, industry, government, foundations. It was called The Future of Biomedical and Postgraduate Training, and we're having a second edition in June. So we are. I can't summarize the whole ideas of the conference, but the purpose was to generate new training modules. How do we need to change our curriculum and the way we approach graduate and postgraduate training to adapt to the current opportunities and realities in basic research? So I'd say one thing, just to echo a comment that Elio made earlier, is, or maybe it was Michael, that PIs should not have the sole responsibility of training their graduate students or postdocs for the breadth of really great careers that are out there, because our experience by definition is quite narrow. So I think institutions are going to need to invest in professional development offices that serve both their graduate and postgraduate trainees, so we can better expose and prepare our trainees for a range of careers, whether it be an academic position like ours, or at a primarily undergraduate teaching institution, in tech transfer, in consulting, in communication, working for a foundation, policy, intellectual property, and law. I mean, there are a wealth of opportunities for PhDs who have excellent training in critical thinking. But it's absolutely true that the PI can't and shouldn't bear the responsibility for that. So I think the institutions are going to need to invest in more career counseling and professional development. And the institutions that get out in front of that are going to be competitive for the best trainees, because you all know the graduate students coming through the doors are well aware of this problem. They read the papers too, and they are, a fewer of them are interested in our jobs. They're really interested in solving big problems and being creative about how they use their intellect and their training. So that's one point.
Michele, don't you think there's also a problem in that the investigators tend to be a little bit, or let me say it, a little bit snotty about alternative careers?
Oh, of course. They have a vested interest in having their trainees at the bench thinking about their very exciting research. Absolutely. So we need to solve that problem. And one of the recommendations is that we move more toward, as you said, staff scientists, so that a PhD trained postdoc could come and have a professional career with an institution.
But it's part of the problem of the way the training programs are often measured. Often they're measured by how many students are have gone on to become faculty members, rather than how many students are still in positions where critical thinking is essential. And I think that's a flaw in the metrics associated with grading of training grants and grading of the success of a postdoc, because the postdoc is not considered successful unless they have secured that PI type of position. While if that postdoc has gone on into science communications or some other aspect in which they still have to apply their knowledge and critically think, it's not considered by the funding agencies to have been a positive event.
If you're funding them off a postdoctoral fellowship or a training program, but what
Correct.
about a professional staff role on an NIH grant? So I think we have to explore that, change the funding mechanisms.
Yes.
The other problem is that no institutions are very good at tracking the outcomes of certainly their postdocs, but also their PhD students. So we, as a field, lack great data on where our trainees have gone and the many ways that they're contributing to our country and to the world. So I think that's a huge gap.
Well, it's the issue of big data. All you need to do is to mine Facebook, and you can effectively figure out where every one of your postdocs has gone, because most people, talk about their jobs on Facebook. Most folks, you can find, Facebook is the wild card in this discussion that has grown up in the last 10 years
Right.
to a place where big data is possible.
A more efficient mechanism that's been proposed by the NSF, I believe, is that upon admission to a graduate program, a person gets an ID, and that ID number follows them for the rest of their career. So it makes it much easier to track not only where has that person gone, but also we could collect data on each principal investigator, who has been in their group, and what are the outcomes of their trainees, so that as a consumer, a new trainee could say, well, here's the profile of this group. Is that really the path that I want to follow? Or perhaps this group that has trained a lot of people and now they're working in, let's say, biotech, maybe that's a better choice for me.
I think these are devices that are handy. So it's possible to track,
Right.
it's possible to do this. That's fine. The point, however, is that the alternative tracks should be made more available, more desirable, and therefore siphon off people from this insanity of having everybody wanting to do the same thing.
Right. And I wouldn't use the word siphon because it has a negative connotation. And as I said, many of our trainees themselves come to us and they're interested in not my job, but other jobs out there. I just placed a really terrific postdoc with a consulting firm, and he's going to do great at it and hopes that it trains him to one day launch his own biotech company. So, I think those are careers we can all get behind.
One of the things they say at the end, which we didn't touch on, is scientists must become engaged in efforts to communicate with the public and with Congress. And of course, this is what we do here on TWiM and all the other podcasts I do. But I would like to suggest that every NIH grant have a communications component. So right now, if you get an NSF grant, I'm not sure which, all kinds or certain kinds, I know I've worked with a few people who have had these. You have to have a communications component. You have to propose how are you going to tell the public about your work, and they will provide money for you to do that. And I think NIH should do that as well. Because right now, we do our podcasts because we want to. We get no credit or reimbursement or support for them. And very few other people are going to do this unless they can get help.
I think that's a great idea. And I'll just say one more thing. Keith Yamamoto, who was a speaker at our conference and who has been a national leader in this area, has a terrific video on the iBiology site where he makes a really clear case that it should be at the level of the PhD that students are exposed to the breadth of careers. And from the PhD, then they make a decision, do I want to go into law, biotech, foundation work, public policy, and then reserve the postdoc for those who are pretty certain that they want to pursue an academic research or run their own research group, either in industry or in academia. So rather than mentors of graduate students just focus on getting their students the best postdoc possible, why not reserve the postdoc for those students who really are interested in an academic career.
Yeah, I like that, but I think exposing them to options at the PhD level is not so easy, right?
And also may be too late.
It's going to take money.
Yeah, because I can tell people about alternatives, but how do they really learn about it?
Well, so from our FOBGAP conference, we generated a series of blueprints, basically, that build on models that have already been established as experimental programs at a range of institutions, from Stanford on the West Coast to Hopkins on the East Coast. So there are some really terrific ideas out there, and we'll make this publicly available in the next few months.
Very good. Anything else, Elio?
No, I think we give people the flavor of what this document is. I recommend that people read it.
Okay. Yeah, I think it's good. And as I said, it's open access. You can always write us and tell us what you think, right? ASM and the International Society of Chemotherapy are joining together to bring you ICAC/ICC 2015. Coming up in September of 2015, it's the principal meeting for clinical microbiologists, infectious disease physicians, researchers, and pharmacists to get together to improve diagnosis, prevention, and treatment of infectious diseases around the world. So this is a combination of ICAC, which is the well-known ASM meeting, the Interscience Conference on Antimicrobial Agents and Chemotherapy, with the International Society of Chemotherapy. Makes sense to bring them together. Over 110 sessions and 19 courses on major advances in drug development and infectious disease research. In addition to the meeting, you'll be able to earn 31 and a half hours of continuing education, get together with over 6,000 professionals from all over the globe, and explore the latest products and services from over 50 companies that'll be exhibiting their wares at the meeting. You can get discounted registration rates until August 6th, 2015. That's not too far away. So go to ICAC.org/ICAAC-ICC. All right, let's move on to a research paper, and Michele will tell us all about this one.
Yes. This was published on July 9th in the journal Lancet, and it's titled Feasibility and Effectiveness of Oral Cholera Vaccine in an Urban Endemic Setting in Bangladesh, a Cluster Randomized Open-Label Trial. And this is by a large group, primarily at the International Center for Diarrheal Disease Research in Bangladesh. The first author is Kadre, and there are several others, and I'll just read their last names: Ali, Shodhira, Khan, Saha, Khan, Begum, Buyan, Shodhira, Udin, Khan, Shodhira, Rahman, Siddiqui, Asazadaman, Akhtar, Khan, Yu, Siddiq, Saha, Kabir, Riaz, Biswas, Begum, Unicum, Luby, Craviato, and Clemens. And some of the others worked at UCLA, at Stanford, at Johns Hopkins, the Vaccine Institute in Korea, and it was funded by the Gates Foundation. And if this sounds familiar to you, it's because it was widely covered in the press by the BBC, the New York Times, NPR, and it's great news, actually. They were able to mobilize a really heroic study that involved more than 200,000 people living in Dhaka, which is the capital of Bangladesh, and they did a very carefully controlled trial using a vaccine that costs, wait for it, less than $4 per person, and were able to demonstrate effectiveness that approached 50% over a 2 to 5 year period. So it's very exciting, and we'll now back up and go through the paper.
Before you get to that, Michele, is
Sure.
$4 really inexpensive when you're considering the annual income of the average Bangladeshi citizen?
No, but if we consider Bill and Melinda Gates,
so you're suggesting that the resource nations underwrite the distribution of these vaccines in order to prevent the spread of diseases of such significance to the rest of the world. It's money well spent, in other words.
Yeah, I bring the $4 up because often we're looking through the the newspaper and we're reading about great new treatments for, let's say, an hepatitis virus, and the price tag is what, $10,000, $30,000 a year. This is less than $4. So the setting that they chose is urban Bangladesh, Dhaka, the capital city. This is a city of 18 million people. It's the 10th largest in the world, and it sits in the Bay of Bengal, which is a delta region. And that is significant because the pathogen that causes cholera is a waterborne pathogen, and it particularly thrives in estuaries. So the experience in Dhaka in the Bay of Bengal is that they have 2 epidemics of cholera each year, and they follow the monsoons. So every fall and every spring, they have about, on average, 300,000 cases per year in Bangladesh, and they have 4,500 deaths year in and year out. More than 4,000 people are dying, and children are especially susceptible. So the disease itself is marked by a profuse watery diarrhea. Patients can lose between 5 and 10 liters of fluid. So severe dehydration occurs, which is marked by dry tongue, sunken eyes, low blood pressure, and if not given oral rehydration therapy, which is basically sterile water with some salts and glucose, people can die, especially children. So this is a terrible problem. Worldwide, there are 3 to 5 million cases per year and hundreds of thousands of deaths. So what can we do about this? Well, fortunately, we know a lot about this pathogen. It's actually kind of humiliating as a person who earns a good income studying mechanisms of pathogenesis to admit that we know in just beautiful detail how this pathogen causes disease, and yet thousands and thousands of people are still plagued by the disease. So what we know is that this pathogen lives, as I said, in natural waters. It's motile. Out in the environment, there are more than 200 different versions or serotypes of Vibrio cholera, but not all of them encode the kind of hallmark virulence trait, which is called cholera toxin. This toxin, interestingly, is encoded by a phage that can infect and deliver this toxin gene into a particular strain. So the 2 strains that have caused most of the disease over the last hundreds of years encode one of 2 different versions of a surface marker called LPS, lipopolysaccharide. And if the Vibrio also encodes the toxin, those are the ones that can cause this terrible diarrhea. And they do that because the toxin gets into cells, and it actually adds a sugar onto a regulatory protein that controls the activity of an enzyme called adenylate cyclase. This enzyme is able to generate a lot of cyclic AMP, which is a small second messenger that controls the activity of other pathways. And in this case, it can control the pumps that regulate salt flux across the membranes of the cells that line our gut. And in particular, when cyclic AMP is high, these ion pumps are dysregulated, so chloride is flowing out of the cells and into the lumen of the gut, and then water follows that ion concentration gradient. So you have water pouring out of the tissues into the gut and causing this huge profuse diarrhea. And with all the fluid loss, there is also a really impressive shedding of the bacteria. So it's estimated that up to 1 trillion Vibrios can be released back into the environment by a single patient. So there's an incredible and impressive amplification of the microbe in the gut, and then it can contaminate the water sources. And in areas where people are crowded and they lack sanitation, where their drinking water and their latrines are not well separated, you can have just explosive outbreaks, epidemics, and even pandemics of this severe diarrheal disease.
Michele, quickly,
yes.
What's thought to be the infective dose? How many Vibrios does it take to infect a person?
I don't know the answer to that.
But it's not a lot, is it?
I don't know how acid resistant it is, but in areas where Vibrio cholera is endemic, and there's not good sanitation, the concentration of Vibrios can get quite high. So that's all I can say to your point.
So, Michele, other than Bangladesh, roughly where else does this occur?
Southeast Asia, it's a serious problem, so India. And then also our listeners may have heard of the just terrible outbreak in Haiti that followed the earthquake. So a classic example is whenever there's disruption to infrastructure and people don't have access to sanitized water or latrines, they're especially vulnerable to spread of this and other GI pathogens.
And Elio, to answer your question, the infectious dose of cholera ranges between 106 microbes to about 1,000. And as Michele was leading, the infectious dose really depends on the gastric acidity. The lower acidity levels reduces numbers of Vibrios required for infection. So if you're on a proton pump inhibitor to prevent acid reflux, it will probably take more Vibrios to give you the disease because the lower the pH, the fewer number of Vibrios you need.
But the point that Michele brought up is that a single patient puts out trillions of blood.
Oh, yeah, 106. So you move the decimal point 2 over, and you still have hundreds of millions of people being infected from one index case.
That's the point I was trying to make. Sorry, Michele.
Oh, not at all. No, I appreciate the discussion. So this study was then designed to take a really challenging setting, which is Bangladesh, and in particular the city Dhaka, and ask whether use of a formalin-killed whole cell vaccine can protect. Is it possible for healthcare workers to distribute this vaccine with public health education campaign to teach children and families to use soap to wash their hands, especially after defecation or cleaning their children's bum, and also a measure to teach the people how to add chlorine to their household water supply to purify it by just adding a few mils of bleach essentially to their water. And then the vaccine that they're using is this whole cell vaccine. So this is a very simple design. It is 3 different strains of Vibrio cholera that are known to be prevalent in the area, and they have just killed these using a fixative formalin and then administer those dead cells in an oral dose. So 1.5 ml, I believe, or no, 1.5 ounces is swallowed by mouth. Luckily, the vaccine doesn't require refrigeration. It's fairly stable. So the design was to identify, well, first they basically stratified regions of this huge city, Dhaka, into 90 different clusters, with each cluster having 3,000 people in it. So this was a massive effort. And each cluster was designed to encompass neighborhoods where people were overcrowded, so more than 3 adults per room. They shared a toilet and kitchen. The water was unsafe, poor sanitation, unhygienic living conditions. So the study design really came up with the most difficult, challenging situation that you could imagine, an urban slum, as they referred to it, to ask if this vaccine, with or without an education campaign, could make a difference.
And the other thing I'd like to add is the study was not necessarily blinded. It was, if you will, in open label, obviously, since they either gave a person a vaccine, and they taught them how to wash their hands, or they did no intervention. So it wasn't the classic placebo blinded type of clinical trial most people think about when they hear the term clinical trial.
And it's kind of an interesting discussion point. We could argue whether it would be ethical to give a placebo. What they did instead, I thought, was really brilliant, which is they appreciated that there is a second pathogen that also causes a similar diarrhea. It's also endemic in the region. It's also spread by water. This is enterotoxigenic E. coli. So they every case of watery diarrhea that came into the hospital, they cultured the stool, and then they used PCR to ask whether this person had enterotoxigenic E. coli, or instead were they shedding Vibrios. So they use that as basically an internal control for specificity to ask whether they saw, the prediction was if the vaccine was effective, they should see a reduction in Vibrio cholera diarrhea, but a steady level of enterotoxigenic E. coli caused diarrhea. So I thought that was a really well thought out design.
And that's one of the challenges that these folks had, is because their outcome is whether or not the vaccine worked. So how do you know if the vaccine worked? Someone has to show up to a hospital to report the diarrheal incident. And the hallmark of cholera, of course, is the large amount of fluid that is generated during the diarrheal episode. So most folks would typically present themselves to the hospital with that, but they did in their discussion address that issue. And the control Michele just mentioned is their way of addressing that particular parameter to know whether or not they did get all the outcomes that they intended.
Right. So they do say that their numbers, their data is probably an underreporting of the success for a number of reasons, but including the ones you gave. But you're right. So the design was, we've got now these 90 clusters with 3,000 people. A third of the clusters, the people are going to get 2 doses of this vaccine. Another third are going to get 2 doses of the vaccine, plus this education to treat their water and use hand washing. And this involved healthcare workers setting up community centers, mobile clinics, and also going door to door throughout Dhaka to educate and follow up with the study participants. And then a third of the participants, there was no intervention. And then 2 weeks after the vaccination, they began to collect records from the hospitals, looking for cases of severe diarrhea that were culture positive for E. coli or PCR positive for ETEC. And they did that at 2 weeks and then followed up 2 years later. And they were able, amazingly, to get 65% coverage. And I say that that's impressive because in this part of the city, there's a lot of migration. So they lost 50% of their participants during the 2-year period due to mobility. People left, migrated for better jobs or for whatever reason. So it's very difficult to track every patient or every participant. And that's one reason they had to use hundreds of thousands of people to get statistically significant data. But the good news is they were able to reduce incidence of illness by 37% with no education campaign, and as high as 45% for those who are also instructed on their water treatment and given chlorine and free soap to wash their hands. And then severe illness, including hospitalizations, was reduced by 53 and 58% for the vaccine or vaccine plus the healthcare education campaign. They also saw evidence for herd immunity, so even people who didn't receive the vaccine were somewhat protected, and that perhaps was due to the education about the hand washing and cleaning water, or because with fewer people shedding these trillion Vibrios into the water, there may have been a lower burden of Vibrio, or lower exposure to Vibrio. That's data that they didn't collect. So that is the bulk of the paper. And again, I just want to say this was a really impressive effort that took a lot of boots on the ground to visit these compounds, to install these water treatment stations, and to teach the people about using soap, especially the young kids. And for it to have been as successful as it was at such a low cost, I think is really now going to put the pressure on government organizations, perhaps local or global, to generate the money needed to distribute this vaccine in areas where cholera is endemic.
Michele, one thing that puzzles me is that cholera bacillus has been known as the agent of cholera since Koch, since the late 19th century. what's so special about this vaccine? Why didn't people do the same thing before? And we take cholera strains, treat them with formalin, and call it a vaccine?
So an earlier version had been used. It was approved in Vietnam, I believe, and they used whole cell killed vaccines, and then they added recombinant cholera toxin subunit B. So subunit B is the subunit that binds to the cells that line our gut. Therefore, if you raise antibody against that subunit B, you can basically generate an antibody molecule to bind and inactivate the toxin. So that had been in use and was somewhat successful, although it required what we call a cold chain. Because that cholera toxin subunit protein is unstable, the vaccine, to be effective, to maintain the cholera toxin antigen, had to be kept refrigerated. So that creates just a huge logistical problem with distribution, especially in underdeveloped areas where the economic resources are limited.
And the magic here, I think, is because it didn't need refrigeration that it showed such promise. I mean,
still, that doesn't explain it. I mean, didn't they, before they knew about subunit B in the year 1900, they must have done this. They must have taken cholera and killed it with formalin and tried it as a vaccine. I don't understand why.
I don't know if it was ingested. I think the hallmark to this vaccine is it's ingested so that when you present the antigens, you're presenting them to the mucosal surfaces, the Peyer's patches, that you're effectively delivering the antigen to the site that the toxin principally acts on. The other vaccines may have been injectables and heat kill injectables, and it goes back to probably our earlier discussion we had on TWiM, where we compared and contrast the efficacy of the oral polio vaccine versus the Salk vaccine, that's the injectable. And I think, Vincent, you were commenting on the differences in the types of correlates of immunity that are elicited from both types of polio vaccine that may speak to it.
Well, they both, of course, make antibodies in the blood, but just the oral vaccine is the only one that immunizes your mucosal surfaces, and that's, of course, where polio infects in the gut. So if you have an immune gut, you won't spread polio after immunization, but you will do so after an activated vaccine. So in this case, this is the place to put it, of course. But, Elio, there have been other vaccines made since the late 1800s.
That's my point.
But I don't know the history. I just know that they did exist, but I don't know why they weren't more widely used.
Well, I think one of the challenges was that they were including the toxin, and so therefore it required refrigeration. So there was discussion about whether this is really practical in regions where cholera is endemic. The second issue is that, and this gets a little bit into immunology, the main antigen in these vaccines is the lipopolysaccharide, which is a sugar that coats the outside of gram-negative bacteria. It turns out that adults and children over the age of 2 can recognize polysaccharide antigen and present it through a pathway directly to B cells that doesn't require T cells. Infants younger than 1 or younger than 2 are not good at that. They can recognize protein antigens, but they can't recognize polysaccharide antigens. So our successful vaccines for pathogens that make a sugar capsule, such as pneumococcus, use what we call a conjugate vaccine. We link the polysaccharide to a protein so that we can recruit the T cell helpers. So this vaccine, interestingly, hasn't yet done that, but yet in this study, they saw effectiveness. Well, actually, they didn't stratify their data for children under 2 and 2 to 5. It was just
under
infants
5
through age 5. So we can't really tell from this data set whether something else on those killed vaccines, a protein, presumably, was actually contributing to protection.
It's useful to look at some of these numbers just to give you an idea. So in the non-intervention group, 80,000 participants, they had 106 episodes of cholera. And then in the vaccine-only group, 94,000 participants in 65 cases. So a reduction, in about 40 cases. And that, when you think about the numbers, it's not a lot of cases, but there are similar issues in testing HIV vaccines. You have huge numbers of people enrolled and relatively few cases. And as a consequence, the vaccine, well, in the case of the HIV vaccine, the latest, the most successful, or the only successful trial, had about a 37% effectiveness, which is at the low end of the group here, the ones which just got vaccine alone without the behavioral change, 37%. So I wonder if this is going to go forward, because for the HIV vaccine, that was considered not useful to go forward.
Well, so they also stratified their data for illness and severe illness.
Yeah.
So the severe illness numbers were better. They were in the low 50%. So with cholera, because once you raise the antibody, you can recover. It's not like HIV. You just need to get somebody through that difficult period, and especially if there's rehydration therapy.
Yeah, right,
if we can reduce the severity, I think that will reduce the economic burden and hardship in these areas.
Yep, good point. How about longevity of the effect? Because I know that these mucosal immunogens are often tough in that way.
Yeah. The good news, bad news is, because cholera is endemic in the region and comes in, after each monsoon season, the thought is that people are being naturally exposed. And so that would provide a natural booster. Now, that raises another issue. One of the controversies maybe around this paper is, should we be investing in this delivery of this vaccine to the populations, or should we instead solve the root problem, which is clean water, access to clean water, latrines, which of course would protect people not only against cholera, but enterotoxigenic E. coli and many other waterborne pathogens.
Yeah.
But we've known about this since the outbreak of cholera in Haiti, and despite our understanding of how cholera got to Haiti, it's still going on in Haiti. And so I think in order to clear up cholera in Haiti, a vaccine strategy in concert with the clean water efforts that many of the NGOs have been doing, as well as the government has been doing in Haiti to control cholera, is probably going to be a way to end the epidemic in Haiti. Because remember, the cholera was imported into Haiti, and if there is indeed herd immunity, if we can provide a mechanism to drop the incidence of transmitting or vertically moving the bacterium around the country, because the Haitians are not subject to the monsoon season and it's not endemic in the water of Haiti, it was effectively an import, we may be able to end the cholera epidemic in Haiti. So as a follow-up, I think the Gates Foundation would look to Haiti to see whether or not we can end it, at least in this hemisphere.
Yeah, actually, there was a study done using the same vaccine in Haiti. It was a report published in PLOS, Neglected Tropical Diseases, in 2014, and they too were able to show some protection. I don't see what the numbers are exactly, but it was certainly a smaller study. And our first author, Firdausi Kadre, was also a contributor to that work, along with a number of labs in the Boston area, Harvard Medical School among them. So this vaccine has been tried in an outbreak setting like Haiti, and now in this really massive trial in a poor region of Bangladesh where cholera is endemic.
Do they indicate, Michele, what's the next step here?
Not in a lot of detail in their Lancet paper.
Lancet's stingy with the word count.
Right.
I mean, do we have to do another trial, or would this be it, and then someone has to just manufacture it and move forward?
That's my understanding.
Yeah.
They had no unexpected safety events.
Yeah.
And they estimated that if they could get about 2/3 of the people vaccinated, they could reduce the total rate of severe illness by at least 40% for at least 5 years.
So Gates Foundation would pay for this, I presume, right?
I don't know that.
Their final statement in their paper, I think, is the most telling. These findings should assist policymakers in formulating rational vaccination programs for cholera in highly mobile, high-risk urban populations.
Basically,
it's a call to arms.
Basically, WHO has to decide, and if they say go for it, then maybe Gates will give them some money.
Yeah. And somebody should calculate what the loss is in hours. It's an economic hardship if the breadwinners are having to miss work, and of course then spreading it to their children.
Right.
But probably back to Elio's point about why is this news when we have had knowledge of the pathogen for so long? I'm not sure anybody has funded a study of this magnitude.
Mm-hmm.
200. They had to start with close to 300,000 people in order to finish with 200,000 and have robust statistical data.
Maybe that's it.
And we still don't have the issue of what the physicists accept as evidence, which is the 5 sigma rule. Biologists only accept a 2 sigma rule. And so the physicists would want a much, much larger study. And in the same issue of Lancet, they're commenting on the 5 sigma rule for medicine.
So what you want to explain that, the sigma rule?
When we do statistics, we've all become aware that if your statistical difference in the null hypothesis of asking the question, are your two populations different, you need a p-value that has a value less than 0.05 or 5%. So you have a certainty that it's greater than random chance to 95%. The physicists look and they constantly are reviewing their data over and over to make certain what they publish is vetted, and they take it out to the 2 sigma rule is standard deviations around the mean. 2 sigmas is 2 standard deviations around the mean, and then 5 sigma would be effectively moving it out 5 standard deviation units from that mean to ask, are the two populations different? So if you would vaccinate a population and have a placebo in another, you would want...
I find this totally irrelevant.
Yes.
Goes for physics, doesn't go for biology. I mean, so what?
No, that's, I think, what was the sense of the Lancet commentary.
Yeah, when are we bothering with what physicists do?
Physicists made an impact on biology, as you know.
Of course, but this is just not relevant. All right.
So a lot of the discussion is let's use this vaccination as one of our tools, but let's not relax on providing clean water and toilet facilities to the under-resourced countries.
Right. All right. Thank you, Michele.
My pleasure.
Very nice. And I would like to read just a couple of emails here before we wrap up. And the first one is one that was passed on to us from Elio. And it's from Nervous in San Diego. Dear professors of TWiM, I was recently introduced to TWiM by a neighbor. For the last few weeks, I have been listening to the backlog of TWiM podcasts, mainly while walking our dog. Regrettably, our dog is a lousy conversationalist. Listening to TWiM while walking is therefore a major improvement. As we walk, the dog investigates things I cannot see, and while I listen to you talk about them. Because I have no background in microbiology, I must deduce the meaning of the 5, 6, and 7-syllable words that you use so effortlessly from the clues provided by the occasional simpler words that surround them. I think it is fair to say that microbiology is the study of what cannot be seen using words that cannot be pronounced.
I agree. I agree with that.
The other day, our dog and I were listening to a TWiM podcast. In truth, I was listening and she was otherwise occupied. You were describing how some parasites modify the behavior of their hosts to the advantage of the parasites. Because I was nearing the end of our walk and the podcast was not yet complete, I decided to take another lap around the block. It suddenly occurred to me that TWiM was modifying my behavior. My decision to extend my walk seemed substantially the same as the behavior of a grasshopper afflicted with summit disease. As I continued thinking about this phenomenon, I surreptitiously checked my mouth for evidence of frothing while resisting the urge to climb a telephone pole. Fearing the worst, my first thought was to rip off my earphones to rid myself of the TWiM infection. But before taking such a drastic step, I decided to solicit your expert advice. Questions. If listening to TWiM is capable of causing me to walk further, is it possible that you are also inducing behavior changes that I have not yet recognized? Will I soon start craving pickles or become addicted to reality TV? Would it be safer for me to restrict my future podcast listening to car talk? I await your response with some trepidation. Sincerely nervous in San Diego. P.S. The weather here seldom changes, but I'll leave it to my neighbor Elio to provide the details. Oh, he's your neighbor.
He's my neighbor, and he's a
That explains a lot.
I would say to your neighbor, watch out for the dreaded symptom of using multisyllabic words that no one understands. If that starts to happen, you've got a real problem.
I don't know if TWiM has become a parasite with our behavior or something alters our behavior. I'm not quite sure. This is a tough one.
Well, if I think it's not a bad one, because if it makes you listen longer, right? Can't be bad.
Walking is good.
Walking is good. I've
Dog
heard.
likes it.
And if you're exercising and you want to listen more, you exercise more. It could be okay.
Are we being sponsored by some organization that promotes well-being?
We should be.
We should be, or maybe an exercise band on your hand. On your hand, one of these new exercise Fitbits or something.
That's right.
Well, I for one appreciate his comments.
It's great. It's wonderful, and thank you for writing. Keep on listening. I think it's interesting that it modified his behavior. Right. So some people, I know there's people who commute in cars. There's this phenomenon that happens with NPR. People get to the end of their commute, they pull into their driveway, and they sit there and wait because they want to hear the end of whatever they're listening to. They're just sitting there, so it's not so bad.
The driveway moment.
Is that the driveway moment? Yeah.
That's the driveway moment.
That's it, right. Yes, okay. I stopped listening to NPR. I listen to podcasts instead. Pavlina writes, hello, This Week in Microbiology. First of all, your podcast is amazing. It made my microbiology preparation much easier in university and got me curious about many things. Now, I've listened to the Battle for Iron podcast, and one thing that I couldn't find on Google got me interested. What is the exact etymology of oceloid? It was mentioned the word has something to do with eye inside of it, and I had to look it up, but nothing really came up. Do you, by any chance, have any idea where it might come from?
I think I do. I had to look it up, of course. Oceloid is something like an ocellus. This is an oid. And ocellus refers to patches of photosensitive pigments, which are not very well organized. So it's not an eye. It's a very primitive proto-eye. And an oceloid is something more organized than an ocellus. So I don't know quite why it was called that, but it's an oceloid is a long way from an ocellus. But that's the origin of the word, anyhow. It has to do with oculus, the origin, the Latin root for eye, for vision.
All right.
Ocular and stuff like that. So it all comes from that. But it's, I don't know. Words come up in strange ways. I don't know why somebody used the term oceloid for this particular organ. But let me just say, a new paper has come up about the structure of the oceloid, and we're going to bring that up again because we love the oceloid.
Mm-hmm. Okay. Anytime. We love it. You're right. Pavlina is from Bulgaria. Nice. All right. One more from Alice in response to the letter from Mark. All right. So I will remind everyone that listener Mark had asked, how do drought conditions change public health risks? California is now experiencing higher incidence of West Nile infections. Should other risks be expected? So Alice writes, besides West Nile and other mosquito-borne illnesses, fungal diseases leap to mind. Dry soil becomes dust that can be inhaled. Coccidiomycosis, a.k.a. valley fever, is probably most relevant to California. There's an open-access systematic review available from PLOS Currents that may be of interest, and she sends a link to that review. So
that's right. But this is becoming a very large subject because there are a lot of things which are being affected. I mean, there is all kinds of things happening which are related to climate change.
Sure, absolutely. In fact, on TWiP, This Week in Parasitism, some time ago, we discussed a paper where they did a study in a region where there is malaria, and they looked at the effect of temperature increase. So usually, as you go up in elevation, so this region had areas of elevated land, in which there was much less malaria because the temperature is lower and the mosquitoes don't persist there. With global warming, the temperature is moving up the mountain, so to speak. So they actually could document increased malaria in these higher elevation regions than previously shown.
Right. All kinds of things are happening.
Yeah.
A big subject.
Hey, that's a good title. All kinds of things are happening. All right. Thank you for those emails. And that's it for TWiM 108, which you will find at microbeworld.org/TWiM. You can also find it on iTunes. You can also play it on your Android or iOS device with the right app, and you could subscribe for free. And if you have questions or comments, please send them to twim@twiv.tv. Michele Swanson is at the University of Michigan. Thank you, Michele.
My pleasure.
Nice to have you back.
Thanks.
By the way, Michele.
Yes?
You sound suspicious, but don't worry. It's okay. I'm not going to ask you to do anything. The last episode, which was just Michael and myself, I actually emailed the first author and got him to tell us a little bit about himself.
Wasn't it fun?
Yeah, it was great. He was really happy to do so. So thank you.
I tried to do that with this week's paper, but they, I'm sure, are just being inundated with media, and so I didn't get a response.
No problem. Elio Schaechter is at Small Things Considered. Thank you, Elio.
I sure enjoyed this. Thank you.
Michael Schmidt is at the Medical University of South Carolina. Thank you, Michael.
Thanks, everyone.
And I am Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Microbiology for supporting TWiM, and particularly Chris Kandian and Ray Ortega for their technical help. The music that you hear on TWiM is composed and performed by Ronald Jenkins. You can find his work at ronaldjenkins.com. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.